and physiologist. This lecture is from chapter 20, and we're talking about blood today. This is part one of this video series. We're going to talk about what blood does, what's in it, and how do we stop bleeding in the process of blood clotting or hemostasis. Our goals when we are learning about chapter 20 are to understand what blood does, what is that function, but also what's in it. And you all know me by now, I like to spoil an ending, is that inside of blood there's plasma, the liquid part, and formed elements. And why are these guys important? We'll talk about blood clotting or hemostasis, and then we'll also talk about blood types and blood transfusions, but that's at a future time. So first things first is what does blood actually do? And there's three main jobs that blood does. So those three main jobs that blood does are one, transport, and then also regulation and protection. But I put an exclamation mark next to transport because blood is still part of the cardiovascular system, and that's such a big role of the cardiovascular system is transport. So what do we transport? There's a lot of stuff. Um, So with blood, we transport things like oxygen, O2 from the lungs. We transport things like nutrients from the GI tract, the digestive system. We transport things like waste, and that waste comes from the cells as metabolic byproducts, and we take them to those places in the body that eliminate waste. And those are, so for like carbon dioxide waste, that goes to the lungs, and then that gets exhaled out. But then we also have, it's called nitrogenous wastes, and those get eliminated via the kidneys. We have units on the lungs, the GI tract, and the kidneys all coming up later in the semester, but this is just like a precursor. But we also transport hormones. And the hormones come from the endocrine system. And the endocrine system, again, talk about it in a future unit. So we transport a lot of stuff in the blood, but we also regulate. And there's a couple of things that I want to tell you about as far as regulation in the blood. One of those things is thermoregulation. So regulation of body temperature. We want to make sure that that body temperature is within optimal range. And so one of the things that happens is that when our cells metabolize, we release heat. We make heat by metabolizing. And that heat is distributed by the blood throughout the body. And so that makes sure that our body is heated. But if we're too hot, then we can cool down by diverting that blood flow to the skin so that some of that heat can be released. So blood plays a really big role in making sure that you're cool enough and warm enough, especially when it comes to getting too hot or too cold. The other thing that we regulate with the blood is pH, how acidic or how basic we are. We have a lot of things in our body that need to be maintained at a constant pH. If our pH is thrown off, then that really interferes with cell processes because a lot of our cell processes are done by proteins, and proteins are a little bit finicky when it comes to that pH. And so with the blood, we have a lot of buffering systems. Buffering systems. And what I mean by buffering systems is that we kind of make sure that we're kind of regulating out that pH. And we'll talk about buffering systems a little bit later in the semester. With pH, the blood also has a ton of bicarbonate ions. Bicarbonate ions, so that we can balance out some of those really acidic products in the blood. So we regulate the temperature, thermoregulation. We regulate the pH, but we can also regulate the fluid volume. We can also regulate the fluid volume, which means we want to make sure that there's enough fluid in the blood to sustain blood flow, but not too much. Okay. And what helps maintain this blood volume is plasma proteins. Plasma proteins help make sure that there's enough fluid and make sure that we're not losing too much fluid because they maintain colloid osmotic pressure. So plasma proteins, they help maintain colloid osmotic, which we discussed in that bulk flow part of the capillary exchange video. Okay, so the last thing that blood does is protection. And with protection, there's two major things that we do. One of the things that we do for protection is preventing blood loss. Prevent blood loss. We want to make sure that we're not losing too much blood at one time, and that's when blood clotting comes in. Which basically means that when you get a paper cut, you don't just bleed forever, because then you could lose blood over time. But lastly, we also prevent infection. And this is done by things like white blood cells, antibodies, etc. And again, we have a whole immune system lecture that we will cover how this works in more detail. Okay, so now that we've discussed what does blood do, let's talk about what's in blood. So what we're looking at here is this picture is called the major components of whole blood, meaning that if we were to take a blood draw like we did in this picture, put it in a vial, and then separate those contents of blood by weight. And this is done in a centrifuge, which is basically like it spins really, really, really fast so that all the heavy stuff settles at the bottom and all the light stuff settles at the top. So this is what our tube would look like after we've centrifuged it. So when I think of blood, I'm always thinking about blood in terms of it being, so blood equals cells plus liquid. That's what I always think about. Blood is cells in liquid. And those cells, they have a special name. They're called formed elements. And that liquid has a special name. That liquid is called plasma. So when we're looking at this little picture here, the first thing that we can see, the top layer is like kind of this yellow stuff called plasma. That's the liquid part of the blood. And notice how much of it there is. It's like over half of the contents of blood is plasma. And then the bottom two layers, so this white layer and this red layer here, those are the formed elements. So those are the formed elements there at the bottom. The biggest one is called red blood cells or erythrocytes. So erythrocytes or RBCs, red blood cells. The skinny white layer in the middle is called the buffy coat, and that consists of two different cell types. So draw this out. The buffy coat consists of leukocytes, which we can also call white blood cells, WBCs. and platelets. And platelets are actually cell fragments, like little bits of cell. And note that this word platelets sounds really similar to plasma. So just be mindful that those words are similar, but they're really different. Platelets are cell fragments, plasma is the liquid part of the blood. So plasma takes up about half of that whole blood contents. Erythrocytes or red blood cells take about a little less than half, 45% or so. That's a lot. And then that buffy coat layer, the leukocytes and the platelets, is like less than 1%. So it's really not contributing a ton, but those white blood cells are doing a lot of immune functions, and platelets play a really important role in blood clotting. So very, very important there. Okay, so let's talk a little bit about some of those characteristics of blood. We can call um, blood is more viscous, so we can say that blood is viscous. It's thicker, it's stickier, and it mostly is because of its formed elements. But mostly the red blood cells, the erythrocytes. That makes blood be a little bit sticky. And if you've ever like tasted blood, you've had like a cut and it got in your mouth or whatever, you know that blood tastes a little bit metallic, and that's because there's a good amount of iron in there. But that other part of blood, the liquid part, the blood plasma, is mostly water. So plasma is mostly water. But there's other stuff in it. There's stuff like nutrients and gases and hormones and wastes and proteins and electrolytes. There's a lot of stuff in it. So there's electrolytes. That's a big one in plasma. Electrolytes make up the most of those contents of plasma. And electrolytes are things like sodium, potassium, chloride, that kind of thing. There's also plasma proteins. Plasma proteins, proteins that are in that plasma liquid. Plasma proteins are typically made by the liver and they hang out in the blood. And if you check back at our bulk flow capillary exchange video, we talk about this a little bit. The biggest or the most abundant plasma protein that I can think of is called albumin. Albumin, again, made by the liver. But it contributes the most to colloid osmotic pressure. Colloid osmotic pressure. So that really contributes to that colloid osmotic pressure. That's that sucking pressure that promotes fluid staying in the blood. By and large, the plasma proteins, their content stay mostly the same. The liver can make more or less of them depending on how much we need that colloid osmotic pressure. But on average, we're thinking about the same. So this is a summary table from your textbook talking about the composition of plasma. So you can see, like I said, there's electrolytes in there, like sodium, potassium, that kind of thing. There's also those plasma proteins. So the biggest one is albumins, but there's also other ones in there. So one of the plasma proteins are called globulins. And some of these are like, again, made by the liver or other stuff. And they can bind to different substances or they can make antibodies. So gamma globulins are antibodies. And then the last one I'll talk about in a little while is called fibrinogen, which is the precursor to some of those clotting factors that we really care about for stopping the flow of blood when vessels break. On this slide, we can see what's going on in the blood, which is kind of a lot. So in this picture here at the top, we can see that we have red blood cells, erythrocytes. So those are the reddest ones. We have some white blood cells, the leukocytes. And then we have these little bitty cell fragments called platelets. So you can see that platelets from a far way away, they might start to look like cells, but if we really zoom in on them, we see that they're just little bits. So think of platelets as just little bits of cell. The bottom picture is taken from a microscope, and we can see we have kind of the same thing going on. We have erythrocytes, red blood cells, we have platelets, little bits of cell, but we also have a bunch of different kinds of leukocytes. So there's some different types of leukocytes that we'll jump into later on in this lecture. The interesting thing is that red blood cells, so these are all the formed elements, right? So red blood cells, we call them cells, but they're kind of not true cells. They're not true cells. And what I mean by that is that they're anucleate, which means they lack a nucleus. So anytime that you slap this like letter A at the front of a word, that means it doesn't have that thing. So they're anucleate, no nucleus. Platelets, they're fragments of cells. And then the other thing about formed elements, we call them amitotic. Amitotic, which means that they don't divide. Again, there's that A in front. So amitotic, a mitotic cell would divide, but an amitotic cell does not divide. All right, so more about red blood cells. So what we're looking at on the left-hand side here is a red blood cell, and then if we were to look at it from its side. Red blood cells or erythrocytes, their shape is called a biconcave disc, which basically means it looks like a jelly donut that you squeezed all the jelly out of. You see in your brain how that would like smoosh the middle of it? And so a biconcave disc is like a cell that was a jelly donut and you squeeze all the jelly out. And so it just means that these middle parts both go in, but the edges are kind of sticking out. Red blood cells have a plasma membrane, so we know about plasma membranes, but they lack nuclei in pretty much all the other organelles that we think of. So no mitochondria, rough ER, that kind of thing, like that's not there. Red blood cells, they're basically just full of hemoglobin. So they have tons and tons of hemoglobin. Hemoglobin is a protein. Hemoglobin looks like this. That's hemoglobin. It's a protein, so that means it's made up of a bunch of amino acids and it's folded all up into this kind of like messed up ball of yarn shape. Each of these different parts that you see here, like this brown part, that brown part, that brown part, that brown part, is all different subunits or smaller part of the protein hemoglobin. And there's two different types of subunits, but there's four in total. Okay, so we have two, I'll highlight it. We have two beta globin chains. This little B with the tail on it is for the Greek letter beta. So we have two beta globin chains, and then we also have two alpha globin chains. So here we have 1 and 2 and 3 and 4 subunits all together. But then this red guy in the middle, 1, 2, 3, 4, red guy in the middle of each of those protein subunits is called the heme group. I'm going to highlight it in pink. Heme group. The heme group is the part of hemoglobin, heme, you see, that contains iron. Fe, which is the atomic symbol for iron. So iron is the thing that binds oxygen or carbon dioxide. So think about this. So if you put those two together, then that's hemoglobin. Okay. So heme has the iron, and think about when you leave like an iron thing alone for a long time, it gets rusty, right? Because that's oxygen and iron and the moisture around reacting together. And if you think about like what color is rust, rust is bright red, and so are red blood cells when they have oxygen bound to that iron, that heme group. So that's really cool. Interestingly, though, carbon dioxide can also bind into hemoglobin, and actually, a good amount of the carbon dioxide in our body is moved around in this way. So the next question is how do we make red blood cells? And the making of blood cells, so making blood cells is a process. It's called hematopoiesis. Oop, I swapped my E and my I. I-E. So hematopoiesis literally is Latin or Greek or something for blood making. And this occurs in red bone marrow. In red marrow. Think back to Bio 430. We have red marrow mostly in our axial skeleton, but also in like our shoulder area, in our shoulder bones, and in our pelvic girdle and like the tops of our femur, so like our hips, hip area. So once our blood cells are made, they enter the blood through these really leaky capillaries. So we make the red marrow makes the blood cells. And then the blood cells enter the blood through what's called blood sinusoids. Those are extra leaky capillaries that have big holes in them so that new blood cells can enter the bloodstream. So if you want to know more about sinusoids and what kind of capillaries those are, check out the blood vessels videos. Okay, but all blood cells, they share a common ancestor, and it's this guy. It's called a hematopoietic stem cell. And we can abbreviate that to HSC, hematopoietic stem cell. So a stem cell is super cool because it has a potential to become lots of different types of cells. And then once they divide, they get more and more specific as they divide until they become like, for example, a red blood cell, an erythrocyte, or any of the other blood cells we'll talk about. But they all come from this guy, hematopoietic stem cell or HSC. So what happens is our HSC will divide into this guy called a proerythroblast. We call that proerythroblast a committed cell, which means that it's not a stem cell anymore. It has a determined fate. It says, I will become an erythrocyte eventually. So that proerythroblast will undergo a series of divisions until eventually we are an erythrocyte, a red blood cell. And the steps that we take in this process of, I'm going to write it in bigger letters, erythropoiesis. That's red blood cell making. red blood cell making. So what happens in this series of divisions is that we're accumulating hemoglobin along the way. So first we divide into the cell that has tons of ribosomes. Ribosomes make protein, which is great because we want hemoglobin, which is also a protein. So then the next phase means that we're accumulating tons and tons of hemoglobin, but also getting rid of all of our unnecessary organelles. The type of blood cell that's actually released into the blood is this precursor to an erythrocyte called a reticulocyte. So right a little bit before we become an erythrocyte, we'll release reticulocytes into the blood, and these reticulocytes actually mature into red blood cells in its very early days of being circulated in the blood. So this whole process, start to finish, takes about 15 days to complete, give or take. So it's really interesting because we can take how many reticulocytes that we have in our blood at any one time and see if it's within a normal range. And if that count is off, there can be something that we may need to look more into. So if we have too many reticulocytes, maybe we're making more erythrocytes than we want to. And similarly, if reticulocytes are low, maybe our erythropoiesis is too slow. We're not making enough of them. And so that could be something that we care about, clinically speaking. Okay. So how is erythropoiesis maintained? So this figure is about regulating erythropoiesis. How is that done? Through homeostatic mechanisms. And we have the same 5 steps we've been talking about. And the punchline here is that it's hormonally controlled. That hormone is called erythropoietin, or EPO, EPO. That's a hormone made by the kidney and liver that causes erythrocytes, red blood cells, to mature more rapidly than they normally would. And this is usually done under what we call hypoxic conditions. Hypo means too low. Oxic refers to oxygen, so that's when O2 is too low. So when oxygen is too low, and a lot of this happens because when we have enzymes that rely on oxygen to work, that means they can't do their job. And when they can't do their job, they can't send signals. When they can't do their job, they send signals to the kidneys and the liver to release this hormone. So when our oxygen-based enzymes can't really work that well, then we start to release EPO. So then we have our 5 steps that we're used to. So that stimulus is that hypoxia, and hypoxia can lead to a bunch of stuff that we don't want. It leads to decreased red blood cells, decreased hemoglobin, decreased oxygen availability. This is no good. So then what happens is the next step, step 2, is the kidney. The kidney and the liver a little bit release EPO, erythropoietin. And erythropoietin stimulates the red marrow to mature those red blood cells faster. So that leads to increasing our red blood cell count, and then our oxygen increases again. So then we're no longer hypoxic. And then similarly, the opposite of true. If there's too many red blood cells, there's more oxygen than we need. So then we would suppress EPO and slow down red blood cell production if the opposite were true. So for the last part of this video, we're going to talk about the life cycle of red blood cells. So from start to finish, what is the life of a red blood cell like? So when red blood cells are made, they do their thing. We undergo erythropoiesis. Those new erythrocytes enter the bloodstream, are deflated jelly donuts, so cute. But these guys are really only good for like 120, 100 days, something like that. But then they start to age out. They start to get damaged. They're no longer good at their job. And so they start to really get kind of stuck in certain areas of the body, especially the spleen. So they start to get stuck in areas like especially the spleen. And so what happens is those old red blood cells, they start to get eaten up by specialized cells called macrophages. So macrophages in the spleen, and your textbook calls the spleen the red blood cell graveyard, and I think that's, kind of a cutesy way to remember it. So the red blood cell graveyard is the spleen, and that's where macrophages eat them all up. And hemoglobin, it gets split up into its parts, and some of it ends up as this molecule bilirubin. Bilirubin. And it's this yellow pigment, bilirubin is, that binds to plasma protein albumin. So bilirubin binds to albumin, um, and then it gets sent to the liver and then to the gallbladder. So bilirubin kind of builds up in the gallbladder, and the gallbladder is this little guy that hangs out right under the liver. So that's the gallbladder there. And then the gallbladder sends all of that bilirubin to the intestine. So bilirubin is secreted into the intestine, and then the intestine is like the last stages of digestion and waste removal. And so what happens is we'll take away whatever nutrients that we can, put it back into the blood, but then what's left is this brown pigment called so this pigment is brown, and it's excreted into the feces. So all of that is the life cycle of a red blood cell.